Okay, let's dive right in. I want you to think about the computers we all know and use every single day. You know, the laptops, the phones in our pockets, they've completely transformed our world. For decades, their power has just been exploding, right? Following this incredible, predictable path of getting smaller, faster, and cheaper. But that amazing era, the one that basically defined the modern age, might actually be coming to an end. So, what's been driving all this progress? Well, it's our knack for shrinking silicon transistors down to sizes that are almost impossible to imagine. But here's the catch: we're getting really, really close to a fundamental physical wall, the atom. You just can't make a component smaller than that. So, it creates this huge question. If we can't shrink silicon anymore, what in the world is next for computing? Well, the answer might not be in silicon at all. It might be hiding in the very building blocks of life itself. The first approach we're going to look at is, well, it's pretty radical. It's about treating the living cell like a programmable machine and biology as a brand new kind of software. This whole revolutionary idea has a name: synthetic biology. And you can think of it like genetic engineering on steroids. It's the discipline of actually designing and building new biological parts, new devices, new systems, right from scratch. Essentially, we're talking about writing software for cells to get them to do new and useful things. What you're looking at here is the biological machine that synthetic biologists want to get their hands on. This is the central dogma of biology. Information flows from DNA to RNA to protein. So if you can design custom DNA sequences, you can control which proteins get made, when they get made, and how many. It's really just like a programmer controlling the flow of information inside an app. But look, to program biology, you can't just poke at it and hope for the best. You need proper tools. And by that, I mean you need real programming languages. We're seeing this big shift away from older, kind of clunky methods toward these really sophisticated, modular languages that let scientists design complex biological circuits in a structured, intuitive way, a lot like how a software engineer would do it. And this is a perfect illustration of that whole design process in action. On the left, you've got a simulation of an initial biological circuit, and the output is, well, it's a mess. It's noisy, it's chaotic, it's not working. But with these new tools, scientists can simulate, test, and then refine their genetic program. And on the right, boom, you get a clean, oscillating output, exactly what they wanted. This is the design, build, test, learn cycle, but for living cells. Okay, let's totally switch gears. So far, we've talked about programming the cell as if it were a piece of software. But what if we use the molecules of life themselves, not as something to be programmed, but as the actual computer hardware? So the core idea is all about how DNA works. You've got your four bases, A, T, C, and G. And the rule is simple. A always pairs with T, and C always pairs with G. This predictable, specific pairing is a lot like the ones and zeros of digital computing. The whole idea is to harness that molecular property to actually perform calculation. A really key mechanism for making this work is something called DNA strand displacement. It's this super elegant enzyme-free way to create logic gates, but at the molecular level. So imagine an input strand of DNA acting like a key. It finds its matching lock on a bigger molecule, binds to it, and in the process, it literally kicks out another strand of DNA, and that's your output. That output can then go on to trigger the next step in a whole computational cascade. So, if we can program cells like software and build circuits out of DNA hardware, what kind of new computer could we actually build? The potential is, it's truly mind-boggling. But as you can probably guess, so are the challenges. The single biggest advantage of DNA computing is something called massive parallelism. See, in a digital computer, calculations happen one after another, just really, really fast. But in a test tube full of DNA, you can have 100 trillion molecular calculations all happening at the very same time. The sheer scale is almost impossible to wrap your head around. To really understand the scale of the challenge here, let's look at a famously complex problem. The traveling salesman asks for the shortest possible route that visits a set of cities and then returns to where it started. It's notoriously difficult for conventional computers to solve as the number of cities goes up. And here is the reality check. To solve this problem for just 62 cities using a DNA computer, the sheer mass of DNA you would need would be more than the mass of the Earth, many, many times over. It is astronomically, physically impossible, and this highlights the central paradox. The very thing that makes DNA computing so powerful, its ability to explore every possibility at once, also creates a completely insurmountable physical limitation.
And beyond just the sheer scale of materials, there are some pretty big practical challenges. You have to design trillions of unique DNA strands so they won't accidentally stick to each other. The lab work itself is incredibly delicate and super prone to error. And maybe most ironically, you still need our classic silicon computers to design the DNA inputs and to read the final results.
So all of this brings us to the future, and what this paradigm shift from silicon to biology really means. It is about so much more than just building a faster computer. It's about a whole new way of interacting with the world. You know, we're at the very beginning of a new field of engineering. For a century, we've engineered stable, predictable, inert materials, things like steel, concrete, and silicon. But now we're starting to engineer systems that are dynamic, that can self-replicate, that can adapt. The rules are fundamentally different, and so are the possibilities.
This isn't just about faster computers. It's a really profound question about the future we're all building. The transition from the Stone Age to the Iron Age changed humanity. The Silicon Revolution defined the 20th century. Well, the mastery of biological engineering could define the next one. What will that world look like? That's the challenge and the opportunity right in front of us.